It had very good PK properties, very low protein shift, very good microsomal stability, very good aqueous solubility and PK. And when you see this in this HDI mouse model, what you see is that at a very low dose, a 3 milligram per kilogram dose or 10 milligram per kilogram dose, it is about 1.4 to 2.5 log reduction in HBV DNA. That's compared to the previous molecule, AB506, which required a 100 milligram dose to get about, or at a QD dose to get about 1 log. So much more potent, met all our criteria. It also addressed the issue with resistance. So we were concerned about T33N and I105T, I105V. And what you see now, we're now in the submicromolar range, very nice submicromolar range, and all the other viral variants that are known to exist for protein, we're in the low nanomolar range. So we're very good now against a number of these variants as we go. PK was very good. Because it's a liver disease, we wanted a very high liver to plasma ratio, and we were getting that with these molecules. And it was very clean in a toxicology sense, no AIDS, no mitochondrial toxicity, no HERG, etc. So this molecule is taken into a clinical trial. First, the single ascending dose, and then multiple ascending dose study, and then into HBV patients. And what we saw in the healthy volunteers, single dose, very nice dose-dependent exposure over time. T1 half about 15 to 20 hours. And then we also saw in the MAD a very nice dose-dependent exposure for this molecule. The molecule was taken in HBV patients, and we were happy to show in a nice dose-dependent manner we got a greater than 3.5 log reduction in HBV DNA. So this is best in class. Shown to be at the time. Now, so we had another little snag here, and that snag is in one of the patients at day 28, so these are only 28 days of dosing, so this is only a phase 1B study. One of the patients showed an ALT flare, absolutely at day 28. It's not a major flare, it was a grade 2 flare. And so we aren't sure again whether this is drug related or is it disease related. So now we're going back and we're doing a 28-day healthy volunteer study with this molecule to rule out any drug related toxicities. So that's where that stands. So we're hoping that, and that should be finished by the end of the year, and hopefully we'll be able to progress this molecule. So now let's turn to S antigen. So S antigen, as I mentioned, is produced in very, very high quantities, and it doesn't have any enzymatic function, so you can't inhibit it with an inhibitor of any type. And so what one needs to be able to do is inhibit its production. So one thing to know about HBV is that HBV only produces one enzyme, and that's the viral polymerase. So there are no other enzymes you can target. So these become very challenging drug discovery projects because of that. How do you address targets that don't have enzymatic activity? And so, one of the things that we want to do is inhibit the production of this S antigen. And one way to do that is to use small interference RNA, so siRNA. So we developed an siRNA strategy to address this. Now, what's nice about HBV is that it's got a circular genome, and it has overlapping reading frames of all its proteins that it produces. And so you can target one place on the viral genome and knock down all the viral proteins that are produced by the virus itself. And so we developed this sequence, modified sequence of RNA. It's got 2' methylated RNA, 2' fluorinated RNA, some phosphorothioate linkages in it, etc. But we also built this, and this is a GalNAc conjugated linker technology. And so what we wanted to be able to do is target the liver. So one thing to know is the liver is covered with acyl-glycoprotein receptors that recognize N-acetylgalactosidase. So it's the liver is pretty exclusive in that sense. The kidneys have a little bit of acyclic receptors on the surface, but nowhere they're not in the liver, and most other cells do not have them. So what we developed is this tetraintegrary ligand that's covalently linked to the sense strand of the GalNAc, of the siRNA. And the way this works is you subcutaneously give the agent, it traffics to the liver, it attaches to the acyclic receptor by the GalNAc units, gets incorporated into endosome. The endosome then releases it, and what happens is that the antisense strand of the GalNAc conjugated siRNA then gets connected to the RISC complex, so it is known as the RNA-inducing silencing complex, that then binds the viral RNA and clips it and causes degradation of viral protein. So this molecule worked very good against low nanomolar, against all HBV variants, all genotypes, so we're pretty happy about that. And in an HBV animal model, known as the adeno-associated viral animal model, we were able to demonstrate in a dose-dependent manner that we saw very significant reductions here, about 4 log reductions in HBV S antigen levels, right, that persisted over time. So this is a single injection, and you see a persistence here almost out to 10 weeks. When you look at the liver histology, what you can see, this brown, if you can see that, is S antigen staining, and you can see in the treated animals, you see no S antigen staining observed, especially in 3 and 9 milligrams. So this went to the clinic about 2 years ago, and what we demonstrated was that, in fact, at a 60 milligram once a month or a 60 milligram once every 2 month dose, we're able to achieve a 2 log reduction in S antigen levels. In fact, once you stop treatment at 48 weeks, these suppressed S antigen levels seem to persist out now for over a year. So we're able to now achieve that reduction in S antigens. We have 2 pieces of the puzzle now. One is the ability to completely suppress viral replication, and the other one is to reduce S antigen load. One thing I also mentioned is that, and I didn't show the data here, but we've done a lot of immune profiling in patients. And what we've been able to demonstrate that those patients that achieve sustained reduction, not complete loss, but sustained reduction in S antigen, you see the beginnings of reactivation of immune cells. Okay, so this is leading to this confirmation of the hypothesis that S antigen is playing an important role in controlling the host immune response. But the other thing we really want to do is be able to boost this insulin, because what we want to be able to do ultimately is give a combination therapy that ultimately will not only cure more patients, right? Because currently, long-term nucleoside therapy for 5 to 10 plus years only cures about 3% of patients once they take the risk of getting off nucleoside therapy. But we want to be able to cure a large percentage of patients in a shorter duration of time. So we want to be able to do all these things. So one of the things we believe you need to be able to do is now boost the host immune response. And one of the things known about HBV is that checkpoint blockade seems to be a thing, checkpoint axis seems to be a thing to be playing a big role in how this virus controls the host immune response. And that is, so if you look at the PD-1, PD-L1, what you see is that PD-L1 expression is upregulated in HBV-infected patients in the hepatocytes themselves. You see PD-1 upregulated in HBV-specific T cells and B cells. And so what's happening is that you're not allowing, you're basically putting the brakes on the immune system by this checkpoint phenotype. A clinical study was done several years ago by Gilead Sciences, where they took a therapeutic vaccine, they took nivolumab, which is Opdivo, a small non-therapeutic dose. This is, nivolumab is a PD-1 antibody, used extensively for treating cancer, a number of different cancers. But they used it here in the study. What they saw was that they had one patient who actually ultimately achieved what they call functional cure. Now, functional cure in HBV patients is loss of HBV DNA, loss of S antigen levels, and the rise of S antigen antibodies. So they're basically functional cured. That's really, they don't lose the virus. The viral DNA, the CCC DNA still resides in the nucleus of the hepatocytes, but you now have immune control over the virus itself. We've done a lot of preclinical work that showed that PD-L1 inhibition with S antigen reduction causes an increase in activation of immune response in animals. And this is the production of interferon gamma, which is an indication of T-cell activation and response. So there's some data, obviously, pointing to the possibility that this would work. Now, we're very interested in this, but we don't want to use antibodies. We actually want to use small molecules. And the reason why we want to use small molecules is because antibodies have this very long, durable effect going on. And as if anyone knows anything about checkpoint blockade therapy for cancer patients, there are a number of significant adverse events that arise from that. Because you're basically, blocking that axis throughout the body. And so what we wanted to do was really selectively target the liver itself. And we believe a small molecule will allow us to do that. It would allow also oral dosing. We could target the liver, therefore we would minimize any safety association with off-target or on-target exposure elsewhere. We also believe that we could tune the effects very effectively using PK-PD relationships. And we think we also would have better tissue penetration, since obviously the liver is a very large organ. You need to get to all aspects of the liver itself. Now, if you look at the literature, there's been some work in this space for non-antibody related PD-L1 binders. Origin has done a lot of work in the peptide space, so these rather modest-sized peptides. BMS has done a tremendous amount of work historically, although I don't believe anymore. They developed these cyclopeptides that are really quite potent, right? They actually had this low nanomolar, the sub-nanomolar IC50 activity, and had very good low nanomolar activity against the CMV recall, which is the functional axis. And then they also identified these small molecule agents, these substituted bi-aryl systems. They went on to demonstrate that these bi-aryl systems. One thing is that these simple bi-aryl systems were really potent against binding. So the HTRF assay, you could show binding inhibition, but you actually did not see any functional activity. It wasn't until you actually begin to grow the size of this molecule now that you began to engage functional activity. It was also demonstrated that, in fact, it wasn't a one-to-one binding that you were seeing. In fact, you were seeing a dimeric binding, so one inhibitor to two proteins. So we became interested in this, actually, when we saw some of the early work in the smaller molecule series, and began to do our own work in this space, and develop molecules that again, grew the size of the molecules, because Jack can get co-crystal structures. We did a lot of co-crystal structure work here to show what the binding mode was like and what was the availability for other interactions. And what you see here is that you have this central bi-aryl core, which seems to be somewhat invariant. It's really important. And you then provide other, sort of hydrophilic interactions at the distal ends of these molecules. And what you also see is that the binding of these inhibitors to form these dimeric proteins are anti-parallel. It's really unusual because it wasn't known in the checkpoint blockade field anything about dimerization of this protein. But this then led us to build, looking at the interactions, this hydrophobic core area and the other hydrophilic interactions that are required, ended up building this pharmacophore model. So the pharmacophore model requires a diaryl thing, which no one has been able to get rid of, a pi stacking unit and the basic groups at the end, for key interactions. And so we've undergone extensive SAR development on each of these positions to ultimately identify a clinical candidate I'll speak to. But one thing we did actually at Arbutus is we were very curious about this dimerization and what was happening. Was it, because it was hard to conceptualize how this was occurring and how the inhibition was proceeding. So we did a lot of work, flow cytometry work, that what we began to notice in fluorescent tagging work was that PD-L1 is on the surface of the cell. Okay. When you give an inhibitor, what happens, you get dimerization, and then you get internalization of the surface of the cell surface receptors into the cell itself, and degradation of those PD-L1 receptors. If you wash out the drug over a period of 4 days, you get reemergence of new synthesized PD-L1 protein on the surface of the cell. So it's a very interesting mechanism by which this, and we published this in Nature Communications last year. So we took this molecule, AB101, it's a very potent inhibitor, IC50, 1.9 nanomolar. We also showed function activity. In fact, it showed to reinvigorate HPV-specific T cell responses ex vivo. So patient HPVs, PBMCs from chronic HPV patients were taken, stimulated with HPV peptide, core peptide, and then inhibitor, and demonstrated that, in fact, equal to an antibody, atezolizumab here, shown here, AB101 can show activation of those immune cells. One of the other things we were very interested was liver targeting or liver centricity. We wanted a drug so that we wouldn't have peripheral exposure, and these drugs had very high 24-hour liver levels shown here. Now, at the time, when we are close to building one, there was no immune HPV model that one could test an immune modulator against. So we ended up reverting to an oncology model. This is the MC38 mouse model, tumor model, and we wanted to compare the ability of this molecule to reduce tumor size relative to a known commercial drug, atezolizumab. And in fact, what you see here, at a 10 milligram QD dose over 28 days, you see equipotent reduction of tumor growth in this mouse model. So then the question is, what about the combination? So we had an immune modulator, we had an S antigen reduction. If you can reduce both, is that going to really boost that potential cure? And so we ended up doing this in vivo. This is a pretty extensive in vivo model study. So we're looking at a mouse model, reduce the antigen load first to reduce that overhang, and then you bring in your immune modulatory PD-L1 inhibitor to see what happens. And what you see is HPV-specific T cell activation happening. So interferon gamma and interferon and IL-2 are increased relative to those that do not have the combination in their association. You see increase in the number of HPV CD8 positive T cells, S-antigen positives, and correlated antigen positive, and you see increase in production of anti-S antibody. So all very positive markers that you're now getting immune reconstitution of. If you look at the antiviral effects, you see the reduction in antigen load, primarily associated with the siRNA, and then you see this sort of plateauing effect with the combination of the checkpoint blockade agent. If you get a second injection at 28 days of the siRNA, you see a further reduction in both HPV S antigen and HPV. So, where are we? So chronic viral hepatitis is a major public health issue whose impact eclipses that of many other infectious diseases globally. The discovery of oral, short-duration, curative therapies has transformed the outcome of individuals suffering from HCV. It has the potential to eliminate HCV globally. But the development of curative therapies for those with HBV is focused on combination approach, as with all other antivirals, with the three key aspects of viral persistence, viral replication, viral replication, tolerance of antigen reduction, and immune activation. So we're very close. Still a little ways to go. It's taking us a little longer than we had hoped in the HBV world, but it's a much tougher problem than the HCV. So I want to acknowledge all those, first those at Pharmasset who did all the work that I showed, except for the phase 3 clinical work, which was done at Gilead Sciences. This whole team is a small group of chemists, did all the medicinal chemistry, all the process chemistry, the process work, the development of the new methodology to make chiral phosphoramidase was all done by this group. Is now used to make multi-metric tons of drug. Great group of people, and then my team at Arbutus, who did all the HBV work and deserve the most credit for everything that I presented. And thank you for your attention. Happy to take any questions if anyone has any. Yeah,
where's the mic? Here, for me, please.
Dr. Sofia, congratulations for your outstanding talk. As we do not have question and answer step after this opening conference, I suggest that those who have some questions to Dr. Sofia to contact him during the symposium, okay?
Sure.
On behalf of the organizing committee, I would like to thank you very much for this excellent talk, and I would like to hand over the certificate of your conference. Okay. Please join me to thank Professor Michael Sofia once again. Just to finish this section, I would like to invite everyone to the social dinner that will be served in the restaurant of the Palace Hotel. Thank you very much.